me and Dan and David and Patrick and Andrew Kaplan, who's not here, looking at SNPs that are related to smoking. So before we just sort of jump into talking about those papers, I think it's important to ask this question at the outset: why might structural modeling be particularly relevant for researchers interested in social science genetics? Why is there a kind of a considerable amount of time today spent talking about structural modeling as opposed to regression discontinuity design or something like this? Why? What is the kind of the connection? So I think that there's a few reasons. One, the first is that structural modeling is useful at sort of suggesting or allowing you to estimate from kind of many moment conditions the pathways that particular variables might operate through. And so this is super important if you're thinking about trying to estimate the causal or the sort of behavioral pathway through which a particular gene operates. And both of the papers that we're going to talk about today have this really as a central question. And I guess we'll wait till each of those applications to talk about the possibilities there. But FTO, which is what Pietro's paper is going to look at, is a good example because this was a SNP that was discovered through a GWAS. And initially, the functional significance of it was not exactly known. Right? It was not a candidate gene. It was something that came about through a GWAS study. And if that's true, right, then you're kind of in uncharted territory for thinking about why a particular gene might be influencing behavior. That if the biology, and there are many situations where obviously the biology is very informative as to what the behavioral pathways might be, but there will be some situations where you're in uncharted territory where you have a gene outcome association, and it might be the case that some of these social science econometric tools might be useful in suggesting mechanisms that are at play, right? So I think that's something that's kind of important. Another reason why getting at the mechanisms is so interesting or important is that you can start to think about personalized medicine, personalized interventions that respond to particular genotypes. Right, that if we know, for example, that a SNP operates through a particular facet of smoking, whether it's through an addiction process or whether it's through preferences for nicotine, then observing someone's genotype and having the knowledge that this genotype works through a particular behavioral pathway can potentially inform the kinds of interventions that you would think about. Secondly, there is also a benefit that goes not from structural modeling to the analysis of genes, but the other way around, from the observation of genetic data to the enrichment of existing economic models. So if you thought for a moment that you didn't necessarily care all that much about genes on their own, but what you care about is building a good model of smoking behavior or building a good model of human capital accumulation, one of the choices that you almost always make or always face is how much heterogeneity you add into these types of models. So for example, if we take a look at this setup for the smoking model, this is a very stripped down primitive model in many respects that we've already talked about. And there's one that we haven't talked about, which is that everyone in this model right now is pretty much the same, right? In the sense that we think about the world, there's tremendous amount of heterogeneity. Some people are just never going to smoke. They don't have any taste for it. Even if you gave them a cigarette exogenously, they would not do anything with it and not enjoy the experience. And some people are going to enjoy it and smoke a lot and potentially develop an addiction, something like this. There's fundamental heterogeneity in terms of how people's fundamental parameters might be situated. These structural parameters might not necessarily be homogeneous. They could be heterogeneous in the population. And if we want to develop a good model, not to understand genes, but just to understand smoking behavior, it might be really useful for us to be able to observe factors that induce heterogeneity in some of these parameters. So on the gene side, on the genetic side, we might care about the gene and ask what pathways it operates through. And then on the economic side, you might say we want to be able to observe factors that might shift around these parameters and give us kind of some empirical meat to hold on to as we try to estimate a model with rich heterogeneity. And if we don't observe those things, if we don't observe, say, individual SNPs that move around these parameters, we might have to just totally integrate out that heterogeneity and kind of assume distributions of these parameters, and that's sort of layering more and more unobservable structure on the problem, and it may be difficult to identify those distributions of unobserved heterogeneity if you don't have some observables like genes to hang your hat on. So I think that there are benefits that kind of go both ways between economics and genes and genes and economics, and so I hope that that's enough of a kind of motivation. So there are very few examples of structural work that incorporate observed genetic variants. In fact, we're only aware of two, Pietro's paper on FTO and obesity, and then our paper on smoking. And so I'm just going to sort of go through each of these papers and sort of provide a background. We're going to, as you might expect, I'm going to spend more time on our paper than on Pietro's, but it's because I'm more familiar with it, and I have lecture slides that I can copy and paste. So I'll start with Pietro's paper, though. So Biroli, 2015, what does he do? Well, he's going to examine the impact of a variant in the well-known FTO gene that's related to obesity. I'm sure everyone in this room probably is aware of this, but individuals with at least one well, I don't know if you know the exact details of this, but you've certainly probably heard of FTO as a gene that's associated with obesity. And the empirical finding is that individuals with at least one A allele in this particular this particular location are at a higher risk for obesity. And so Pietro is going to, first of all, replicate this in a particular data set in the ALSPAC data, the Avon longitudinal study of parents and children, where he's going to get, mothers and young children. He's going to observe these kids at young ages through kind of teens and early adulthood. And look at how obesity outcomes evolve, and also look at how food intake evolves, also look at how exercise evolves. All of the kind of big inputs that you would expect to function in a model looking at obesity. Obesity and BMI, and this is sort of getting at this mechanisms issue, it offers a great example of a genetic association which could be rationalized through multiple mechanisms. So one, if I knew nothing else about the biology, and there might be someone here who knows more about the biology of obesity who might absolutely shoot me down and say, oh, well, the biologists know exactly what's going on. If that's true, then that's my fault. But I don't know that, and I don't think Pietro referenced it either, so I'm assuming that there isn't much clarity on this. We could imagine that a SNP that's associated with obesity could be rationalized through a couple of different mechanisms. First, we could imagine just a preference for food, a preference for caloric intake. It could be that some people just are going to choose to consume more, and they get more utility out of consuming calories in every period, and that's what might rationalize a kind of rise in obesity by genotype, a gap in obesity by genotype emerging over time. Or, secondly, it could be that this gene affects the process that governs how food inputs are converted into weight, or converted into adipose tissue, converted into obesity over the life cycle. It could affect metabolism, in other words. And it's not clear ex ante which channel a particular SNP might operate through. And so what Pietro is going to do is have a model where genes can affect both this, well, he'll call it a production function for obesity, a sort of mapping between current choices and future obesity, or does it come through kind of preferences for food itself? So the very first thing, just to give you some sense of the magnitude, the scale of the association that we're talking about here, if you look at body mass index at different ages, age 8, 11, and 13, looking separately for males and females between the genotypes that have at least that have 2 Ts versus those that have at least one of the risky A alleles, what do you see? Well, there's definitely there's an association. It's on the order of about a 2% increase in body mass at each of these ages. So you see between the T allele and the risky allele, there's definitely an elevated BMI for individuals that have this sort of risky A allele. It appears to be a bit larger bigger of a gradient for women than for men. And they're also, it appears, I mean, there's kind of mixed evidence just in the descriptive statistics about whether they're more or less likely to be sort of sedentary. You could ask this question of is the gene might be operating through the difficulty of exercise or something like this, and so the Pietro is also showing you how number of sedentary hours per day are affected by this as well. And there's kind of, it's hard to interpret really what's going on there. It seems to be elevated levels of sedentary activity for men and women, although at age 11, or excuse me, age 13, it kind of closes. If you look at the evolution of body mass, sort of a picture is worth 1,000 words maybe, you can see these kind of gaps, although kind of non-existent or very slight in early childhood, and then about at sort of age 6, these gaps start to emerge, and they widen over time, and maybe they ebb and flow over the life cycle, but certainly from age about 6, 7 onwards, you see the kind of gaps that emerge between people with the A-risky allele and the TT genotype. Okay, so some very descriptive evidence. So Pietro's paper is going to do sort of two things. That he's going to show some patterns in the data with just some basic regressions. And I'll look at if I take BMI on the left-hand side and I add a bunch of regressors on the right-hand side, I'm going to get some coefficients that suggest some patterns. And I'm not going to go into a lot of the details of the estimation method because his paper is a bit more is a bit different than the two kind of methods we talked about. It's a kind of indirect inference calibration exercise, but you can think about it for the moment that he's going to have some descriptive regressions, and he's going to ask the simulated model to generate regression coefficients from this table that look the same. And so what are these regressions? Well, in the first column, just looking at the relationship between log BMI, that's the dependent variable, and having the risky genotype. And there, as we said, you see from the descriptive statistics about the same thing. There's about a 2% increase in body mass if you're of this sort of risky genotype. You can start to add in some more covariates that we think might be relevant. If you add in lagged BMI, obviously the contemporaneous impact of the gene goes down because presumably the variant is also affecting your lagged BMI, which is very strongly associated with current BMI. And then he starts to add in food intake, sedentary minutes in a day, and then an interaction between the gene and these two things. So if you start to sort of pick apart and say, how am I going to possibly pin down the importance of one mechanism over the other? Well, if it's simply a matter of more food, right, then what should we see? We should really see that it all comes through just the effect of food. That once I control for food intake, we would think that the effect of the gene might be greatly diminished, or that it might not appear. So if we add food intake, obviously the amount of food you consume is positively associated with BMI. But there's still some role for this SNP. In fact, if anything, compared to this specification, it goes up, although it's not statistically significant. And then what? Well, then he can also look at interactions between food intake and sedentary minutes and the gene. And this would maybe get at trying to pin down whether the gene is operating through just a level or if it's interacting with the choices that people make. Is it a preference for food, or is it a difference in the way that the same amount of food intake gets mapped into obesity or adiposity later in life? And so what do you see there? Well, the interaction between this genotype and food intake, he actually finds a positive and significant interaction. So for the same level of food intake, if you have a different genotype, that's increasing obesity at a faster rate than people who have the less risky genotype. Interestingly, you don't see the same set of interactions with sedentary time. So if you can, seems to be ruling out a mechanism that goes through the efficiency of exercise or something like this, that there's no interaction between sedentary minutes and obesity and the gene. Okay. So what do we kind of see here? What is a very rough takeaway in these descriptive regressions? It would seem that there is some effect of the FTO gene even after you control for inputs like food intake or like exercise choices. And there also appears to be a role for some interaction between those choices, at least food intake, and the genotype as well. So to cut to the chase, I mean, he's going to eventually find evidence that at least the way that his model is set up, that he can't reject the hypothesis that FTO is working through both of these, although he's going to find that it seems to go through a preference for food much more than this other mapping. So let's, yeah.
Yeah, I happen to be the person who knows a bit about
Great. So certainly, yeah.
And I'm just wondering, there's a problem in the field in general, not just this paper, that the food intake measures are often underreported, particularly with children.
Mm-hmm.
That is, people either forget what they ate, or they just, I don't know why they underreport, but like 60% of people report they consume less food than they should, if you calculate how much they exercise. And interestingly, this trend is correlated with BMI, which is higher. People with higher BMI tend to underreport more.
As someone who's obese, I can tell you I know exactly what's going on. If someone asks me what I ate yesterday, I'm definitely going to lowball it probably, and going to have more shame about it than someone who is 20 pounds lighter. So I had exactly the same thought, sort of, in response to his paper. So it's that certainly is plausible, and that is potentially something that would generate this descriptive pattern, not just an actual interaction in the production function, but reflecting this idea that there's some systematic misreporting that's growing in obesity, and therefore also growing with the genotype. That's absolutely a possibility.
And the second problem is that depending how the sedentary minutes is measured, and again, well, there's some studies claim that the self-report activity questionnaires are essentially junk. Because people, similarly, it's not because of shame, people just can't remember. Or particularly, they have no time minutes, how much time I ran, or how much time I cleaned my room, like human beings.
Yeah, right.
So, and what measures that are slightly better are just like these kind of personality type of measures that did I do less or more. And this has some concordance with actual, like, accelerometer measured activity. So in a way, I think the sedentary measure would also have quite a little reliability. And I wonder how this would affect the model.
Well, so one way, let's get back to this after we see his estimates, but you see kind of where we're going with these two separate mechanisms. One way that would affect the estimates is that there could be a bias in the direction of this production function mechanism, right? That it could be the case that there isn't any real interaction between the gene and the food intake and creating obesity, but it's just this pattern is an artifact of reporting. Obviously, if you ask the simulated model to match these patterns and you don't have shame-induced reporting or something in this, the way the model is going to match it is by loading onto the production function, right? The model, those estimates, if that's what's going on in the data, the model estimates are going to be biased in the direction of that story, because that is the only way that model has of fitting that pattern of the data. So that's definitely a source of bias.
Yeah, it's not just a shame. I mean, there are other mechanisms. Some people just say that bigger people have bigger portions, and because it's a 3D estimation, the bigger portions are hard to estimate for everyone.
Mm-hmm, mm-hmm. So
it's not necessarily the same path.
Okay, I'm calling it that because okay. Yeah, so this brings up a larger point too of how do you handle, how inside of a structural framework would you want to handle measurement error? So Ken Wolpin, who's in labor economics, is one of the big, and Michael Keene are sort of two of the figures that are most associated with structural methods in labor economics. In many of their papers, they sort of add measurement error processes where they say, I simulate the data, now I have this simulated data set, but then I can add some noise on top of it. I can add measurement error to the underlying true simulation data that my model generates. And so that way, I can kind of match the process that we think might be going on in terms of the way that these variables are misreported by survey respondents, et cetera. I don't know how that would look tweaking Pietro's model, but you could think about doing something like that. But we'll think about that more later. Okay. So from these reduced form patterns, we can figure out a structural, relationship between the FTO variant and deeper parameters involved in the process of generating obesity. This first requires being explicit about what model you think is generating the data that you observe. So Pietro's model is, if you're a health economist or you're aware of this, it's very closely tied to a canonical model called the Grossman model. And basically there, what did Grossman do? Grossman basically said in the same way, that you had these kind of models of savings behavior, where you have a stock of savings that you can invest and there's some rate of return and it grows over time and you can consume out of that stock. You can potentially model health in the same way, that there's a stock of health, which could be very abstract, and that you make investments in this, either through exercise or through eating right, doing these sorts of things, and this stock can grow or it can depreciate over time, and the size of this stock is what's related to the health outcomes that you actually experience. So like in all of these models, the goal of the individual is to maximize the present discounted value of lifetime utility, recognizing both the short-run benefits of health and the short-run costs of investing in it. So what are the kind of, and you'll see too that most of these models have a kind of common structure, right? There's a period utility function. There's what are the benefits that you get each period. You have to make choices about what goes into your utility. You have to make choices about functional forms for these things. Okay, then you have to specify how are the state variables of the system evolving. The variables that I care about that I track over time, how are they being impacted by my choices? So this slide, we're just going over those two important things. So in this model, the flow utility that I get in a given period, it's going to be a linear function of a bunch of logs of outcomes and choices that people make. So these zetas are coefficients on the logs of some important values. You get utility directly from BMI. Presumably zeta B is negative. There's some disutility as your BMI increases. Now obviously at very, very low levels of BMI this would be the opposite, but we're dealing in a developed country context where we're not going to worry too much about undernourishment. There's also the utility that you get from food, F, food intake. Now notice that zeta F is allowed to vary by genotype. So basically he's going to estimate two separate versions of zeta F, one for people that have the risky allele and one for people that have the less risky genotype. Then there's utility that you get from leisure, zeta L. It multiplies log of leisure, and then there's utility that you get from the consumption of all other consumption goods beside food, C, and zeta C is the coefficient on that. Yeah. L, we'll get to that in just a second. So there's gonna be time. Every moment, every period t, an individual has a time budget that they can spend that time either essentially exercising or enjoying leisure. And so the more time that you have, the more time that you have to sort of exercise. There's a trade-off. This basically captures the idea that it's costly to exercise. Right? It captures the idea that I have to give up something if I spend an hour at the gym, and that something that I give up is time I might enjoy doing something else. This equation is run off the page, but not too much because it just ends right there. So you can you're not missing anything. The second important equation is how does BMI evolve? And so here, again, something that's linear in logs. Log BMI is going to be a linear function of what? It's going to be a linear function A of just a baseline intercept that's determined by genes. It's going to be a function of your food intake. The coefficient on log food is allowed to vary by genotype again. A function of exercise, that's allowed to vary by genotype, although we have kind of descriptive evidence that would also already suggest that this probably doesn't vary too much by genes. And then we allow, this should be bt plus 1. There's a little bit of a typo there, that this is log bt plus 1 as a function of everything in period t. And then obviously there's persistence in obesity, persistence in BMI, and so log bt affects log bt plus 1 and in a way that varies with time. So this is kind of a nonlinear specification that allows the relationship between current BMI and future BMI to evolve differently as the individual ages and t increases. So importantly, notice that FTO little g enters in a number of places, including the marginal utility of calories and the relationship between food and exercise and future BMI. And really that's going to be the kind of estimation story. What can we infer about these things? And so if you wrote down the optimization problem for this model, again, it's going to look a very similar structure we had before. Every period, the problem is to maximize with respect to the two choices, food and exercise, period utility, plus what? The discounted value of future utility, the expected value of the value function that you're gonna get from waking up next period, period t plus 1, with a BMI of bt plus 1 and an epsilon shock of epsilon t plus 1. This is the evolution equation that we looked at before. Oh yeah, I guess I did miss, there's something, you did miss something. You missed this epsilon t, which is a random component to the evolution of BMI. Here's the budget constraint. You have a certain amount of income each period that has to get split between food and other consumption goods. The price of food relative to C is PF, so the more that you spend on food, the less you have available to spend on consumption. And then here's that time constraint issue, that every period there's some amount of time that you have omega that can be split between leisure or exercise. Now, this doesn't feature prominently in this paper, but it is a prominent feature in the original Grossman model, is the idea that health impacts the amount of time that you have available for doing things in your life. So the idea is that if you're in bad health, you're spending time taking care of yourself, you're sort of incapacitated, you don't have the same amount of time to devote to other endeavors, including preventative care for yourself in the form of exercise here. So don't worry about that for now. This doesn't play a big role in Pietro's paper, but it is something that's in the original Grossman model. This kind of idea that the amount of time you have to do stuff is a function of your health. Yeah.
Is the exclusion of savings, is the exclusion of that have a negligible impact on the actual results?
I don't know. The short answer is I don't know. We could think about what that might do. And this is going to be one of these problems in a lot of these models where if you have a continuous state variable, like for example BMI, you usually want to kind of avoid adding another continuous state variable if you can. And so very often the choice that people are going to make is with something like savings is to sort of give up on that unless there's some reason you think that this is really strongly interacts. One way that I think you could imagine it interacting is if there were ways that you could purchase healthcare for yourself in the future. So if I have a risky health outcome right now, what are my options right now for dealing with that? Well, the only preventative measures I can take are to make sure that my obesity doesn't grow too much, right? You could imagine a world, however, where my ability to save would be an alternate mechanism for me to deal with the health consequences of obesity. If I had enough monetary resources, I could afford better health care, I can deal with the health problems that arise. That might affect some of the behavioral estimates. It's not clear. I mean, it's an open question, but certainly this is another example. These are the kinds of questions that you're always going to have to deal with these sorts of structural models, that if I don't add this mechanism, am I biasing my results? If I add it, am I going to get something different? And it's important to think through why the inclusion or exclusion of a mechanism like that might affect the parameter estimates that you get. Okay, so the results, the actual specific parameter estimates are not all that terribly interesting, but there is going to be a decomposition exercise that I think is a useful takeaway. The estimation of the model suggests that FTO affects both preferences for food and the relationship between caloric intake and obesity. That's something, and it's no mystery how we're getting that, right? It's no mystery why this structural model suggests that, because it's being asked to match data that sort of pretty clearly showed that in a descriptive analysis, right? That FTO was impacting obesity and it was interacting with food intake. The decomposition exercise indicates that about 35% of the effect of FTO is due to differences in the production function parameters, how food and exercise affect future BMI. About 65% is due to differences in inputs, food intake, and exercise. Okay. Why then is this potentially interesting? Well, I think that's interesting in its own right, but what you can do now, remember, this is one of the big benefits of of structural estimation is that once you've estimated a structural model, you have a little miniature world, right? You have a little snow globe that has little people running around in it, responding to shocks, responding to any kind of crazy policy you want to cook up and you want to throw at them. And you can see how the people in your estimated model will respond to those policy changes. So those are referred to as counterfactual exercises. So Pietro is going to go through two separate counterfactuals and try to look at how FTO might have impacted differentially people's responses to these changes in the environment. So the two counterfactuals are a 50% increase in the price of food and a 25% reduction in food intake during the first 10 years of life. So this would be kind of just an exogenous, whatever you choose, you're not going to be able to consume as much. So this, you might think of this as something that a parent might enforce upon their child, or you might think of this as something that a school system might be able to enforce by offering less caloric foods and just restricting the choice set in terms of how many calories people might be able to encounter. So this line, remember this kind of evolution, and the numbers. Pardon me, forgive me, I'm going to kind of just eyeball some of these numbers. If you look at the differences, say in the mid-teen years here, it's a difference on the order of about a half of a BMI point, maybe 0.4 BMI points for women, and something less than that for men. The first counterfactual exercise, the 50% increase in food prices. This is now the snow globe is being shaken up, and you're kind of looking at what happens differentially at genotypes. Well, a 50% increase in food prices does something pretty dramatic to kind of cut down the differences. There wasn't much of a gap before age 5, but you see that this gap now is much tinier, right? The left-hand side scale here, right, is half of a BMI point. So these differences are very kind of tiny, and then after 18, there's essentially no difference between these two genotypes in the simulated counterfactual. So that policy results in an average reduction at the start of adulthood of about 0.6 BMI points, but that seems to affect much more the people that have the risky allele than the people without the risky allele. There is less of an effect of this policy of just sort of constricting intake early on in childhood. And so the reason I think for this as well is what are you doing with that type of a policy? That you're sort of reducing the welfare, the utility of the kids, because they would choose to consume more food, right? But you're not doing anything that's going to alter later on in life the incentives that they have to eat more or less food, right? So the thing about the 50% increase in food prices is that it's actively shifting the choices that people with the gene make, whereas if I temporarily restrict that early on in childhood, assuming that the preferences, the mapping between the gene and preferences is stable, as soon as you leave that age 10 environment, the same dynamics are probably going to kick in as well. So these counterfactuals at least suggest that kind of broadly taxing calories might do more to reduce genetic differences in obesity than say an early childhood policy that restricted intake. Again, lots of places you could disagree or poke holes at modeling. It's always true with structural models, but this is one of the few examples we have of someone who's done a very serious job of incorporating genetic heterogeneity into, yeah.
I was wondering what, like in the previous graph, the difference between the alleles kind of merged together after.
Yeah, I don't know.
But in a way, that would tell that the policy is more effective on the other approach, or I don't know.
Well,
Tell what the effect
it is hard.
baseline.
It is. I agree. But this is the presentation of the results in the paper. But it's also this policy. Just the average effect is much lower, and it's not differentially affecting the genotypes. So the conclusion that he's reaching is that the increased prices have a bigger effect on average, and they also have a disproportionately big effect on people with the risky allele. Okay. So shifting gears, this paper with many of the people who are in this room and Andrew Kaplan, who's not in this room, embeds genetic heterogeneity into a structural model of smoking. So again, we're going to look at a different health behavior. And again, one of the most important questions we're asking, like Pietro, is through what channels do genetic variants operate? And so here, Pietro is asking, we have a kind of preference for food, or we have a kind of production function by which choices get mapped into obesity. We're going to be looking at similar mechanisms for SNPs related to smoking. And so what do? We're going to replicate 4 SNP smoking associations in the health and retirement study. These SNPs are associated with unexpectedly large effects on health. That is, we're going to find, as has been found elsewhere in the literature, for example, Van der Wiel et al., that these SNPs have kind of modest impacts on cigarettes smoked per day, but they seem to have larger than expected impacts on lung health. And so this poses a bit of a mystery, or it's at least something that could be rationalized through multiple different stories. And so part of what we're doing in this paper is to say, can a dynamic behavioral model of smoking rationalize these kind of slight effects on observed smoking and these bigger effects on health? We're going to embed this genetic variation into this model. We're going to have kind of 2 channels, essentially. There's a preference for nicotine, similar to Pietro's preference for food intake, and there's going to be something related to addiction dynamics. Is it the case that these SNPs operate how the addiction to nicotine grows, or how costly it is to withdraw after a kind of history of smoking? One thing we're going to do, like Pietro, is look at how, in some counterfactuals, we're going to show that, that differences in these genes might generate differences in how people respond to policies. And I think this is ultimately an important policy point, because it says that that different policies may affect different people in different ways. A lot of difference in that sentence, but that's kind of the point. And you may be left with a world, if you just think of what's been going on with smoking for the last 20, 30 years. You've had steadily rising cigarette prices, right? And it's often an interesting question, who is still left smoking, when you're paying $13 a pack for cigarettes? That presumably the population of smokers today is different than the overall population of smokers in a world where it was much different, right? And so it may be the case that you now have a population of smokers that are going to respond to different policies, that maybe simply increasing taxes, you've reached the limits to what you can do with a policy like that. There might be other options, other channels through which to address them. Now that's an ambitious question that we actually don't even answer necessarily in the paper, but it's something that I think motivates this exercise, and understanding how different genotypes respond to policies could have an important public health consequences, implications. So there are 3 recent GWAS papers in Nature Genetics. Well, recent 10 years ago, but it's recent in the grand scheme of things. Where specific genetic variants associated with smoking outcomes, many of which reside in some kind of well-known chromosomes. Many of these are understood to contribute to the synthesis of subunits in nicotinic acetylcholine receptors, which are sort of channels in brain tissue that affect how nicotine interacts with reward systems. And we'll choose to study SNPs that are associated with an intensive margin of smoking, that the biggest hits, the most robust genetic associations tend to be with smoking phenotypes that are related to the intensive margin, how many cigarettes are smoked per day. There's far weaker associations for the extensive margin, for whether anyone has ever smoked, for example. And so we're going to focus on these intensive margin relationships. There's 4 SNPs that we're going to look at, and there are many, many, many hits, many, many genome-wide significant hits in these GWAS studies. What we did was to sort of focus on the top hits in sort of 4 distinct regions of the genome. We essentially looked we selected these SNPs with sort of in Plink we can sort of calculate genetic correlations between SNPs in different regions. We're looking for the SNPs in each of these regions that had the largest independent signal. And so we selected these 4 before doing any analysis of our own, just from the GWAS metadata. So between 2006 and 2008, we have about 12,000 people in the HRS that were genotyped using saliva samples. An important point to consider here is that the sample naturally belongs to later birth cohorts than the overall HRS sample. So I think this is an incredibly, important point, and it's a point where structural modeling actually offers some advantages and can actually inform better empirics on gene behavior associations. We think in a lot of different contexts about sample selection problems, right? But now think about genetic associations with health behaviors when you're genotyping people who are old. And now think about when the technology became available to actually genotype people, or when tissue samples were collected that were later subsequently genotyped. You had to survive in the HRS until at least 2006 or 2008 in order to be in the genetic sample. Now, that might introduce a particular kind of bias in reduced form estimates of the impact of a particular SNP on health behaviors, right? So think of it this way: if I have a risky genotype, and I might have very likely died before I had a chance to be genotyped. So who are the people that remain in the HRS to be genotyped in 2006, 2008? Well, if I have the risky allele, I might have had other characteristics that made me healthier than the average person. Right? And so that might actually attenuate the relationships between SNPs that you observe in a data set like the HRS and health outcomes that might drive mortality. And in fact, we have some interesting evidence on this. It's very, very suggestive, but I think interesting nonetheless evidence that this is true. Now, what can I do inside of a structural model that could allow me to correct for this? Well, I can just mimic exactly the same selection procedure with my simulated moments. Right? I can say that I'm only going to calculate simulated moments for individuals that survive in the simulated sample to 2006 or 2008. Right? And so therefore I can get rid of, in the same way that the HRS's selection procedure weeds out people that might have been predisposed to die earlier, right? I can weed out the simulated individuals when calculating the simulated moments that would do this as well. And that would generate valid moment conditions. I would again be in a world where the expected value of my moment conditions is equal to 0 only under the sort of true parameters of the data generating process. We're spent a lot of time, so I'm going to not spend too much time on descriptive statistics, but very much what you would expect. We're going to look at a bunch of health outcomes that are sort of pretty clunky. They're not terribly specific, but are very informative nonetheless. One of the most important variables we're going to look at is something called ever-lung. There's a question in the HRS: have you ever had a major non-cancerous lung illness? And specifically, that's either chronic obstructive pulmonary disease or chronic emphysema. There's not, unfortunately, the HRS is in the non-restricted data. We can't identify specific cancers. So even though we have an ever cancer variable, we can't pin down a particular lung cancer. So that's a limitation, certainly, at least of the current version of the data that we're using. But we're going to see there's plenty of interesting action on this ever lung illness variable as well. So the very first thing we do is replicate in a very reduced form setting the associations between these SNPs and the variable, the intensive margin variable that's used in these gene discovery exercises. Now, this is a very particular measure. And this is going to be an important part of our story of reconciling these findings. That because of the need to harmonize phenotypes across samples that have been pooled together, there's often a kind of least common denominator problem. That if I want to get large sample sizes for a particular phenotype, I need to have something that's consistent across many different studies in order to pool them together. And so that means that I might have very interesting longitudinal data on something like smoking that is multifaceted, that has sort of a ramping up of intensity and a sort of plateau and a cessation. And I might not observe that in all of my data sets, but I might observe something like, what's the most cigarettes you ever consistently smoked in a day? Right? And so if that's the best I can do, that's common to all my data sets, then that becomes then the discovery phenotype that you're going to select, because that's what's going to give you the kind of power to detect actual associations. We're going to say later on that might be problematic because it obscures a lot of the interesting dynamics that might be happening over the course of the life cycle. But that having been said, that variable, this maximum cigarettes smoked per day, this retrospective maximum is the intensive margin phenotype that's used in a lot of these genotype in these GWAS studies. It's the one we can also observe in the HRS. and we replicate for at least 3 of the SNPs statistically significant associations that are of about the right magnitude. So we've changed the reference alleles on all of these to be the protective alleles, the alleles associated with reduced smoking in the GWAS studies. And what do we find? Well, for all but one of them, we find statistically significant negative associations, as you would expect. We're going to flag this and keep track of this because we're going to see something interesting with this RS132 SNP a little bit later on. It's important to note that, for reference, RS169 has been found to have an association of about negative 1.3 extra cigarettes per day in Beirut 2010. So just within the HRS sample, we're replicating that almost exactly, and that's from data that's not the HRS, or at least not just the HRS. If you move to lung illness, you say, okay, we have a SNP that's associated with smoking, one endogenous variable. Now we have a SNP that's associated, we might ask, what is the ultimate impact on health? Those are these other endogenous variables in the system. So we're going to have those results in columns 2 and 3, but in column 1, I want you to just focus your kind of the ground your reference or comparison to the reduced form association between maximum cigarettes per day and lung illness. So obviously this is not an exogenous variable, but just descriptively, what do we find? We find that every extra cigarette per day increases the probability of a major lung illness by about half a percentage point, a little bit less than half a percentage point. Now, if you move to regressions where you include the SNPs, what do you find? Well, for RS169, RS132, we find pretty big associations between the SNPs and lung illness. Each extra copy of the protective allele reduces the probability of a major lung illness by about 2.7 percentage points for RS169 and about 2.3 percentage points for RS132. Now, if you thought for a moment of what would you expect this effect size to be, if you had a naive model in mind where you say, what is the impact of the SNP on smoking, cigarettes per day, and then what's the impact of cigarettes per day on health, what would you get? Well, for RS169, we know that's associated with about 1.2 fewer cigarettes. In column 1, we multiply this by 0.004, we should expect an effect size of about negative 0.005. Now, granted, there's all kinds of problems going on with this regression. This is surely endogenous and associated with the error term, and we're aware of all those problems, but at least to benchmark your expectation, that's about what you would expect. And we're finding something that's many multiples of that. That's much larger than you would expect by just looking at the SNP association with smoking and the smoking association with lung health. So that poses a bit of an interesting, that poses a fact that has to be explained. It's something, it's a pattern in the data that begs a kind of an explanation. If you look at mortality, and these are sort of very, very simple, crude, 1-year death rate linear probability models, what do you find? Well, interestingly, if you look at all cohorts, you always estimate a kind of negative association between these protective SNPs and mortality, but they're not statistically significant. But remember, we had this possibility of a potentially substantial selection bias problem here, because the old are particularly the cohorts that had to do a lot of surviving in order to be genotyped. So if you split the sample between older cohorts and younger cohorts, those born between 1920 and 39, those between 1940 and 1959, lo and behold, you sort of see a pattern that's very, very consistent with that story of selection bias, that for the older cohorts, we don't find anything that's statistically significant in terms of death rates. That'd be very consistent with a world in which there is a selection bias and the risky genotypes that survived had other attributes that made them exceptionally healthy. But the younger cohorts, we see pretty substantial negative associations between these protective alleles and mortality probabilities. So this is one of these cases where it says, hey, there really could be a kind of problem here, and one way that you can solve this is by addressing it through a structural model. If you look at the health outcomes related to never smokers, we find sort of goose eggs, across the board. That we don't find any association between these SNPs and health outcomes for never smokers, which strongly suggests that whatever these SNPs are doing, it's going through smoking behavior. It's mediated by actual smoking behavior. So our main SNPs have modest effects on the number of cigarettes consumed per day, but appear to have very large effects on lung health. How can we reconcile these facts? One hypothesis might be that the genes affect the sensitivity of lungs. We think this is inconsistent with the results on non-smokers, although it's difficult to push that too much. It may be that the non-smokers, we haven't observed the kind of lung insults that they might be experiencing, and if we could, we'd want to test whether they're more sensitive to environmental pollution, for example, but we don't necessarily have that data right now. So what are we going to do? So we're going to build a life cycle model, structural model of smoking and health to try to tease out where these SNPs might belong and whether that can explain these kind of patterns that we see. So what do we have? So this model is going to look suspiciously similar to the simple one that I gave you in the other lecture for obvious reasons. But it's going to be a little bit different. It's going to be a little more, a few more bells and whistles, and a little bit different concept of, there's going to be a concept of addiction that's not present in the other one. So time is discrete, and it runs from age 10 to age 90. You die with probability 1 after age 90. Each period, you're going to choose a quantity of cigarettes to consume, 0, 10, 20, and 30. We're choosing these because they're real big spikes in the distribution that we observe. These are real points of mass in the distribution of responses, and lots of responses are clustered around these. And we think this captures light, medium, and heavy smoking to a pretty good extent. We're going to have 2 continuous state variables, so 2 stock variables. And we saw that stock evolving in the simple example earlier today. Well, we're going to have 1 stock that governs the relationship between past smoking and the incentives to smoke. The simple model that we saw in the other lecture had no sense of addiction in it. There was no sense that if I smoke more, it changes my incentives to smoke in the future. It just had this notion that your past smoking impacted your current health. We're going to explicitly build that in the model. We're going to have an addiction stock, SITA, and a health stock, SITH. And these 2 stocks are going to affect different things. One is going to affect how past smoking impacts future behavior. One is going to affect how past smoking impacts future health. Now, that distinction we think is kind of important. For a good example, which is very much tailored to the health outcomes that we observe. With lung cancer, there's pretty decent evidence that cessation kind of makes a difference. That if you quit smoking, that risks of lung cancer fall pretty substantially with the time that you've successfully quit smoking. Chronic obstructive pulmonary disease is actually different. There's actually evidence that this pretty much involves permanent damage to lungs, and that even after someone has sort of quit smoking, they have permanently elevated risks of these sorts of non-cancerous lung illnesses. Because that's really what we measure best in the data, it makes sense to have a health process that accommodates for that fact. That also means that it might be important to allow someone's addiction to die before the effects of their past smoking have died on their health. You have to allow for the possibility that someone could have been done with their addiction, they successfully quit, and yet they still are at permanent elevated risk. Because of that, it's important to have 2 stocks, we think. Okay, there's laws of motion that govern each of these things that look just like the laws of motion that we saw earlier. For the health stock, it's very, very simple. Again, sort of linear. There's a depreciation rate, delta H, on the old stock in terms of producing the new stock, and new smoking boosts the future stock, health stock by some parameter, zeta H. The addiction stock, we're going to have a very similar structure, but we're going to allow different depreciation rates or different mixing parameters depending on whether you are consuming more than your reference point or less than your reference point. Less than your reference point or more than your reference point. So the idea is that your future stock is going to be a weighted average of your old stock and your present consumption, and we're going to allow that weighting parameter to be different on the way up in your addiction versus on the way down. And that just captures this kind of idea that it might be the case that it's easier to develop an addiction than it is to kick one. Right, that you can sort of get addicted to something relatively quickly, and it might take more time for it to die out than it did for it to increase. So health while alive is reflected in a pair of indicators, 0, 1 dummy variables, indicating a chronic smoking illness or some other bad health. We have this 5-point scale in the HRS. Are you in 5 different increments of health status. So this is basically mapping into whether you're responding that you're in bad health. So, so one is sort of overall health, and one is smoking-specific health, which is whether or not you reported a major non-cancerous lung illness. Okay, this evolves according to a bivariate probit specification with age, and the health stock and the path health status as covariates. So I know I'm losing the room. I know that the fatigue is setting in and the energy level is dipping, and that's okay. That's okay. We're gonna get through this. So before the period starts, the following probit specification determines whether or not an individual dies. So on top of the health process, there is a death process, which is exactly the same as what we saw before, that it's a probit specification, and the things that enter the probability of death include age, quadratic in age, these health indicators, and also directly the smoking stock that you've accumulated. So now here's the period utility, and this is basically the last thing to round out the model before we can kind of show the estimation results. What goes into period utility? So there's a couple of things. First term is the utility that you get directly out of smoking. That has some marginal utility, alpha not I. Now we're going to allow alpha not I to be heterogeneous in the population. That is, different people can have different means of this variable, and there's going to be random shocks on this on top of that. We also allow for the utility of smoking to diminish if you're in bad sort of lung health. So there's evidence, that lots of people kind of quit when they experience a major lung illness. Now, some of that could be endogenous health responses, that I know that my health is sensitive at that moment, so I want to quit. Some of that could also be a shift in preferences. Your doctor tells you you have to quit. If you don't quit, you're going to be ruining your health, even if you wouldn't personally sort of respond that way. So we allow for this to capture the big decline in smoking that you see when people get diagnosed with a lung illness. There's log utility from consumption besides smoking. So this is income here, the YIT. This is your expenditure on cigarettes. And now this is the sort of addiction process, which we govern as a kind of cost of doing something, smoking less than your current addiction stock. I'm going to talk about that on the next slide in more detail. There's also disutilities, alpha BS, alpha BO from either of the bad health states. So one of the reasons why I don't like smoking is because I might fall in these bad health states, and that's costly in terms of utility. Now, what goes into this addiction term, or what goes into this reduction cost term? What we're saying is that if, so this is an indicator variable, you consume fewer cigarettes than your addiction stock, then that triggers a flow of negative utility, of disutility. And what is it? Well, it's something that grows potentially non-linearly with the distance between your stock and consumption, right? And there's a random component to it. So this is the nonlinear term, and this is the kind of random component, the exponential of alpha I1, and then some random normally distributed cost term here. And what we assume specifically is that this intercept here, alpha 1, can take two values, a kind of a low value or a high value. So the heterogeneity that we imagine in the population is that there's sort of two types of people. There's one type of person who finds it easier to quit, easier to reduce their consumption relative to their stock, and one person that finds it harder to quit. So that's one form of heterogeneity. There's two types of this alpha 1 intercept that you might have. However, we also allow people to be uncertain about the values of this alpha 1I. And so there's a learning process involved in this as well. Okay, so I'm going to actually skip over the learning process because we're running out of time a little bit. And it doesn't actually function very much in the results. That this learning stuff doesn't seem to really matter in the model that much. So what do we have? Well, we have kind of heterogeneity in the population that can be driven by genes. So either your taste for cigarettes, these alpha not I's, or the addiction type, this alpha 1I, can be, there's different flavors of this out there in the population, and we assume that there's 3 preference types and 2 addiction types. So in other words, what we're assuming is that this alpha not here, that goes into the marginal utility of smoking, that there's sort of 3 flavors for the mean of this. That there's a sort of low, medium, and high value of this, and that you're one of those types and you're getting shocks on top of this. And then for this, alpha 1I, there's sort of 2 flavors of this out there in the population. So there's sort of 6 total types of people. 6 types of people. There's a low cost, high cost addiction type, and then there's sort of a low, medium, high preference type. So there's sort of 6 flavors of people out there in the world. And what the genes do is affect the probability that you're one of these flavors of people. Now, there's many different ways you could model genetic heterogeneity. We could have allowed SNP counts to directly impact alpha. We could have allowed SNP counts to directly impact the alpha 1 or alpha naught. The problem with that, and this is very much related to the earlier comment about the curse of dimensionality, is we have 4 SNPs, right? There's 3 different values that each SNP can take. So that means we have 81 different distinct combinations of each of these SNP counts that we could have. Now, some of those cells are missing, but we have 81 different kind of flavors of people. We would have to solve this model 81 separate times if we wanted to model heterogeneity as being totally unique by genotype. This way, if we say there's sort of 6 types of people out there and the SNPs basically affect the probability that you're one of these types, we only have to solve the model 6 times. Right? So that's sort of one way that you can kind of deal with some of these cursive dimensionality issues, is to assume a kind of simpler structure of heterogeneity and allow something like genes to just impact the probabilities over these different types. Okay. Okay, so what do we do? We estimate this with a method of simulated moments. Here are the actual parameters that we estimate. And usually in these structural models, it's difficult to make heads or tails of numbers like this. So what I'm going to do is instead skip to some pictures. These are the different types, 4 of the different types. And you can see how they're behaving differently as they age. So the left column here, we have people that are the low addiction type that find it easier to quit and reduce smoking. On the right, we have people that are the high addiction type that find it harder to quit. And then within each graph, the different lines represent the preference types, the low, medium, or high preference types. So what do you see? Well, people with higher preference types, what is the expected value of cigarettes per day? Well, it's always higher for the higher preference types. As people age, they cut back and they reduce the amount that they smoke in each period, with the exception of the very end of life, where there's some Mickey Mantle dynamics going on. In all of these papers, right? In all these models, there's always this tendency that as people get close to the end of life, they're gonna smoke like chimneys, right? Because they don't care anymore, because they're gonna die in 3 periods. So there's no costs anymore to any of this. So you always have to kind of fight against that a little bit, or just, we don't really use moments later on in life. We just, these models, I think, are just, those predictions or something else going on very late in life, and a more complete model would maybe allow for the utility to shrink as people get older, but that's something that you very routinely see. If you look at the lower panels on death rates, again, what you would expect, higher preference types have higher probabilities of dying, and all of these things are shifted up for the people who are of the higher addiction type. Okay. How can we map genetic heterogeneity? Well, remember, the goal was to map genotypes into this parameter space, the links between genetic heterogeneity and heterogeneity in these structural parameters. So here, what do we have? X-axis, the expected value of the marginal utility of smoking, the preference channel. On the y-axis, the expected value of lambda, the sort of reduction cost parameter. And each dot here, each circle corresponds to a genotype, and the size of the dot indicates the prevalence in the population. So one thing we observe, there's sort of heterogeneity, right? These dots are not all lining up. Different genotypes are associated with different combinations of these structural parameters. One thing that remains to be explained is that, if anything, there seems to be a kind of negative relationship between the expected value of the marginal utility of cigarettes and the ease of quitting, which is kind of not exactly what you would expect. You might expect that some genotypes may make it harder to quit and harder to and make you like cigarettes more. We're kind of finding that the high preference types seem to have an easier time quitting. We're still in the process of tweaking this model. It's not clear that will survive the kind of refinements, but if it is, it could point to, it could really be there in the world, which would be kind of surprising to us, that a SNP that's protective would kind of be associated with a less protective structural parameter, but it could also be that we're just getting the model wrong, and that it's compensating for the model misspecification with something that's a little wonky. Yeah.
Is the linearity of each 3 frequency?
Well, that's what you're picking up there is a SNP that is not impacting. One of the SNPs is not impacting one of the parameters, right? So you could have multiple combinations. So this, so imagine you had this genotype, and now you change that genotype, right? Just on one SNP, but that SNP doesn't actually impact the reduction cost parameter. Well, that new circle is going to be very close to this, on this dimension.
I guess I'm trying to understand why there's sets of threes that are all linear.
It's related to the fact that one of the SNPs doesn't impact one of the channels very strongly.
I think he's referring to if you look in the kind of the top left.
No.
On top of each other.
Yeah, that's what I know.
So it would be affecting the parameters a lot, right? Both of the parameters a lot if it's creating lines. Isn't that
No, it's not creating lines. It's creating nested circles. That's what you're referring to, right?
No,
Oh, you're not.
So
Yes.
The top left, you have
Yes, that's right.
a linear set of
That's
three
right.
at every single one.
That's right. That's evidence of the flip side of that, which is there's some SNP that's really strongly shifting. There's some SNPs that are very strongly shifting these parameters, generating these stacks. And then there are other SNPs that are not affecting these things, so you get things that are piled on top of each other. When you have, I think this is one way to sort of report this data. You could look at the individual SNP by SNP associations of these things, but there's sort of a lot of those parameters floating around. So this is, I think, an easier way of just getting the point across that we are estimating substantial genetic heterogeneity. So we'll finish out with the two counterfactuals that we consider. What if we reduce cigarette prices in all years by 50%? Or two, what if you delayed the Surgeon General's report until after 1995? So I think this is a good example of a counterfactual that we truly couldn't do, without a kind of structural model. That in the model, and this is something that I kind of skipped over, one of the problems with a lot of structural models of smoking is that they typically ignore the fact that people's understanding of the health risks of smoking evolved considerably over the 20th century, right? That until the Surgeon General's report, the conventional wisdom might have been that maybe smoking wasn't the greatest thing for you, but it didn't become a kind of public health concern. It didn't become the kind of received conventional wisdom that smoking was going to damage your lungs until after these sort of movements in the 1960s. I mean, if you want to think of how much the pendulum shifted, look at some of these cigarette ads from like the 1940s, where you have like doctors saying, well, I smoke, Marlboros because they're the smoothest on the lungs or something like this. There's literally ads that are like this, right, where doctors are giving testimonials for particular brands of cigarettes as being better for your health than others. So obviously you would never dream of having that happen today. So you really was an era where these public health risks of smoking were not really internalized in decision making. So one thing that we do then is we sort of solve two versions of the model. One where people are uninformed about the health risks of smoking, and one where they're informed about them based on our estimates. And we assume that behavior until 1965 is governed by the uninformed model, and then after 1965 people are responding to actual health risks associated with smoking. So what we do in this second counterfactual is just delay that Surgeon General's report until 1995, so about 30 years after it actually happened. And so I think the takeaway that we want to end on is, if you look at either of these counterfactuals, and more so with changes in cigarette prices, is that genetic heterogeneity can induce sort of substantial differences in how people respond to some of these policies. Now, if you looked at something like the kind of 2-year death rates here, there's a big mass of individuals, some genotypes that are down here, and there's a non-trivial pool of genotypes that are experiencing death rates, responses to death rates that are, say, twice, almost 3 times as much as the response that you see for some of the less affected genotypes. Similar, if you just look at binary smoking rates, there's sort of a distribution that ranges from anywhere 2.8 percentage point differences to about 4 percentage point differences. So genes that are sort of impacting maximum number of cigarettes per day in relatively modest fashions can induce non-trivial differences across genotypes in responses to changes in the environment. And I think that if you look at something like the information channel, some of this becomes really pronounced. So if you look at how the information intervention would affect smoking, or the delay in the information intervention, well, these are huge differences, ranging from 10 percentage point differences to 20 percentage point differences, and the kind of mode of around a 17 percentage point increase in smoking rates. So those are big, economically substantial differences in how people are responding to information interventions. And so this really suggests that genetic information can shed some light on how people might respond to policies that you might not instantly think of in terms of targeted policies. Things like information interventions, not necessarily about genes, but about sort of health risks. You could imagine a kind of world where if I know something about your genotype and I'm your doctor, I might want to be very careful to know that you have been presented with salient information about health risks, for example, because I might know that you're the type of genotype that could really respond to this more than others. So that sheds, that opens up some possibilities that this type of genetic heterogeneity could be used in meaningful ways in actual policy applications. Okay, so I think I'm going to end with, yeah.
So is the distribution of treatment effect not necessarily driven by heterogeneity in the shocks that people experience from the simulation of the model? Or
What do you mean?
so like in your model there are shocks like in addition to some type of like addition.
Oh yeah, that's right, that's right.
And that could affect the handwriting type of thing.
You're right. It would be possible. I mean, so I think our same, obviously any of these simulated moments are going to be generated with some simulated sampling error. So it's possible that you get a spread just based on that. In a full proper version of this, we would have sort of confidence intervals on all of these things, and in principle you could calculate that. I would say certainly these enormous differences are very unlikely to be attributed to simulation error. I mean, also the number of simulation runs that we have is I think fairly large. I think we're in the realm of like 30, 40,000. And I just don't think that these counterfactuals are gonna really vary that much based on sampling error. Okay, that's really I think that's a good point to end on. A little bit of a marathon through structural methods, but I hope at least in the second lecture you get exposed to the kind of practical work that people have been doing, trying to bring the idea of structural estimation into the world of genoeconomics. So thanks. After lunch, what I'm going to do is in that session, for those that are interested, kind of walk through the code that I have that solves that very, very simple model that we looked at in the first lecture. So for everyone who's interested and wants to maybe look at what that looks like or what might be involved, please stay tuned for that.